Okay. Hi.
Hi.
I'm Grace.
This is Ava.
Uh,
We're Hell's Kitchen. That's
We're
our
Hell's
group name.
Kitchen, and we're
That's our
gonna
group name.
talk about the virus PowerPoint.
We're making our concept map on the whiteboard back here.
Yes. So we'll start with section 1, which is the search for elusive viruses. And one of the first discoveries was when Pasteur used attenuation to make the vaccine for the rabies virus.
Which was like continuous reinfection.
Yes.
Until it weakened.
And he coined the term virus.
Which is Latin for poison.
Yes. And it was first used to describe any known cause of infectious diseases, but that was changed later. And then as Koch developed his postulates, more and more causes of infectious diseases were attributed to bacteria, but you can't, they realized that it was something smaller than bacteria that was infecting people. I mean, not people, that was causing infections. And
they were going through the filters.
Yeah.
And they were like,
it wasn't filtering through.
That's still causing the infection.
And so viruses cannot be cultured using the same methods as bacteria and fungi. And they were first discovered by filtering infectious fluid through a filter that was designed to trap bacteria. So section 2 is talking about the position of viruses in the biological spectrum. And rather than calling them alive or dead, they refer to it as active or inactive. Viruses are obligate intracellular parasites. They cannot multiply unless they have a host, and it's a specific host.
And once they invade, they take control by regulating the synthesis of more viruses within the host. But they're technically like inactive macromolecules whenever they're outside of the host.
And viruses can infect every type of cell, so bacteria, algae, fungi, protozoa, plants, and animals. Let's see.
They have a basic structure of a protein shell or a capsid that's surrounded by a nucleic acid core and can have either DNA or RNA as genomic material, but not both.
Yeah, that's some properties.
And they lack enzymes, which is a really unique characteristic, and they also lack the machinery for like making proteins, so they don't have any ribosomes.
I guess for under that, we could write DNA or RNA. Yeah.
All right. So
Inactive or inactive. Yeah, we did that.
I that Coke was smaller than you found that they were smaller than bacteria.
Okay. And on that, electron microscopes are required to detect them. They're really tiny. Yeah, talking about the capsid, I think you said it's made of protein subunits surrounding the nucleic acid. And then the nucleocaspid is the caspid and nucleic acid together. Not all viruses have an envelope, but it's only in some animal viruses. Spikes found on both naked and enveloped viruses. This is how viruses attach themselves to the host cells. Difference between naked is that naked viruses consist only of a nucleocapsid. They don't have an envelope. And then, let's see. I guess under structure, just put naked versus enveloped. I don't really know how to say it. I don't know English.
Can
capsid,
all the viral
spikes.
components.
Yeah, I have it right here if you want to write it. Or you want me to call it out?
Capsid,
Envelope, spikes. And then viron, which is a fully formed virus able to establish an infection. Ooh, and then we have the viral capsid shapes.
The viral capsid shapes. Oh.
So like helical,
Yeah.
icosahedral. We already talked in complex. It's helical, icosahedral, and complex. So helical makes a rod-shaped virus. Icosahedral, 3D virus, round. So yeah, it would be round, not rod. And then complex is a mixture of both. Found in bacteriophages, viruses that infect bacteria.
What's that second one?
Icosahedral? Icosahedral? ICO. Oh my gosh.
And then fully formed virus.
We talked about that.
Then we can go into the genome.
Yep.
Which has the DNA carried by the cell, blueprint for everything.
Blueprint for everything. And like we said before, viruses contain DNA or RNA, but not both. Number of viral genes is small compared to that of the cell. And the genes it possesses are specific to how, like, what they need to invade the host. Is that still under?
Yeah, we're still under.
Okay.
You can write. Talk about. No, we can keep going. About reverse transcriptase.
Yeah. Yeah, synthesizes DNA from RNA, which is only found in viruses whose genome is in RNA form, which makes sense. Because it transcribes that RNA into DNA. It's kind of cool.
And then those retroviruses borrow
Yeah.
the tRNA, right?
Yeah. Retroviruses borrow the host's tRNA molecules. Just stealing all our DNA.
Jeez.
So mean. So let's see. Yep.
For classification.
That's these. So it's this whole slide.
The formal and the informal.
Yeah. So informal would be animal, plant, or bacterial virus, enveloped or naked, DNA or RNA, helical or icosahedral, so everything we just talked about. And then formal would be structure, chemical composition, and genetic makeup. And I think that's like the last of. That should be. Yeah, that should be the last of
Yeah,
that. We already talked about bacteriophages.
Yeah, I think
Oh.
just those. There's the two life
Yeah.
cycles. The
Lytic and the lysogenic life cycle. In the lysogenic cycle,
that's where it infects it, right?
the DNA becomes like, hold on. Yeah, it's like